All right, well, good evening, everybody. I'm Ravi Majeti, and thank you for having me. I understand this is a very eclectic audience, so I'll do my best to explain it to the whole group. And what we work on in my laboratory is stem cell biology as it relates to human acute leukemia. Leukemia is a acute leukemia in particular is a very aggressive blood cancer. This is the type of cancer where patients need chemotherapy for about a month. They're in the hospital. In many cases, in order to be cured, they need to have a bone marrow transplant. And I got interested in this field. I'm also a physician and I'm the past chief of the hematology division here. And it's really motivated me to know that the chemo that we primarily give was all implemented in the 1970s, just about the time when I was being born. And it fundamentally, of course, has changed a little bit, but fundamentally hasn't really changed at all in that entire time. And the reason why we're going to be talking about that is there's some new strategies that we've been developing that are bringing some new ideas to the forefront. And this slide is illustrative of cellular reprogramming. We normally think of cells as fixed in their fates. Cells of the heart are fixed as heart cells. Cells of the skin are fixed as skin cells, etc. But there has been over time an appreciation that cells can be more plastic than we originally had thought. And there have been a number of different advances that have demonstrated the direct conversion of one cell type into another. Now, these are just different examples of that and the different types of cells illustrated here. This one, panel C, where skin cells or fibroblasts from the skin could be converted through the introduction of a few gene factors into pluripotent stem cells. And this has been a fundamental discovery and development in stem cell biology. Actually won a Nobel Prize about 10 years ago. But even prior to that was the discovery that in the blood lineage, there are cells of the B cell lineage. B cells could be converted into macrophages, a different type of blood cell, through the manipulation of different key lineage defining factors. So these are transcription factors. So we were interested in the fact that B lineage cells could be converted into macrophages. Now, in the blood system, there are a number of different types of cells. There's lymphocytes that include the B cells and T cells. B cells make the antibodies. T cells do a lot of the killing. We need all of that for our immunity. There are also other cells that are less specific, these macrophages. They're just big eating cells. That's what the name means, macro big phage eater. These big eating cells, and they do a lot of different functions in the immune system, and I'll come back to some of that in a moment. Now, we were interested in this disease, precursor B-cell acute lymphoblastic leukemia. What great names we have in medicine. Essentially, it is the acute cancer, leukemia, of B-cell progenitors. That's what this means. We make B cells and the other cells of the blood in the bone marrow, and they undergo a transition from very immature cells that start with blood stem cells, not pictured here on this schematic, but they go through a series of transitions before they ultimately have all the properties of the mature cells that carry out their functions in the blood. And this cancer is really characterized by a block in the normal development of B cells at this particular pre-B cell stage. So they're almost B cells, but not quite. And then a lot of studies that have interrogated mutations that are present in these cancers have demonstrated that they often have mutations in these key factors that I've boxed here in red that are involved in defining the B cell lineage. And in fact, just going back to this previous slide, this particular factor, PAX5, that when it can be ablated in an experimental system, can allow B cells to convert into other lineages. And in fact, you'll see PAX5 is commonly aberrant or mutated in B ALLs, which led us to hypothesize that these B leukemia cells, maybe they could also be reprogrammed into the macrophage or myeloid lineage. So we set out to test that. This is a set of experimental data that I'm showing from patient samples. These are 3 different patient samples. We collect them from our patients here in the Stanford Hospital, of course, under human research subject rules. And we wanted to test whether they could be converted from this B lineage that's marked by this particular marker, CD19, into that macrophage lineage marked by CD14. So this is a type of data where there are many different individual cells being analyzed, and they pile up into these contours, this contour plot. And over time, we found that, yes, some of these cells could be converted into this macrophage-like population. And they have various other features of macrophage-like cells that aren't so important to note, other than that they also have expression of different types of markers that are involved in key functions of macrophages. They look like macrophages under the microscope. They do the types of cell killing that macrophages do as part of the innate immune system, and that's what's highlighted on these particular schematics. So what we wanted to see was whether these cells, we called them macrophage-like cells or MLCs because we couldn't say that they were definitively macrophages, but whether they still had the potential to form leukemia. And so that's the experiment that's described here. The experimental system we use is we can take patient cells and transplant them into specialized mice that lack immune systems, so that human cells will grow in the mice, similar, although not exactly the same as they grow in humans, but that's the animal model that we can use. And so if we take the patient cells, that's the blue, directly from the patient and inject them into these mice, over time, these mice all die. These are survival curves showing days on the x-axis and the percent of the animals surviving here, so that they all die, the blue curve. These are two different patient samples. And we can do our little culturing trick to try to convert some of those cells into the macrophage-like cells. And still at the end of that culture, we still have some leukemia cells. And we can collect those at the end, and that's the green, and they still cause leukemia and kill the mice. But in red, what you see is these macrophage-like cells, they no longer have leukemic properties. They don't kill the mice. They can't make leukemia. And that was really cool. We were converting leukemia cells directly into another blood lineage cell that lacks cancer-forming properties. Now that was an exciting and an unexpected observation in many ways, but it's not a strategy to really bring to patients because, as you can even just see from this schematic, only a portion of the cells, not all of them, 60+% in this particular example, would convert, and you're still left with a bunch of leukemia cells in the patient. But that brought us to another idea that relates to how the immune system can be trained to target cancer. And that's what's really shown on the left. This is a complex set of experiments that had originally been done by investigators here at Stanford and ultimately formed the first type of cancer vaccine. And this was about 20 years ago. And the key to get a cancer vaccine is the idea is very different from your COVID vaccine or your other vaccines against infectious pathogens. Those vaccines are designed to give you immunity in the setting where you don't have the disease yet. That way your immune system can be trained ahead of time so that if you encounter the pathogen, you'll have immunity against it. Cancer vaccination is not that. Cancer vaccination is trying to get the immune system in a patient who already has cancer, but for the immune system to then recognize the cancer as something foreign, just like we do a pathogen, and thereby fight off the cancer, kill off the cancer, because the immune system is exquisite at getting rid of something that shouldn't be there or that it's trained to recognize. But the immune system, as you have to think about it, can't do that in an unregulated way. It has to do it in a very highly coordinated way. Otherwise, we get autoimmunity and all sorts of other problems that come with the immune system being activated in the wrong way. And when I say the immune system, really what I'm talking about is these T cells and B cells that I mentioned before. And T cells in particular are the most effective killing component of the immune system. And they are very tightly regulated. Not only do they have all sorts of different types of control, but they can only become activated by a specialized cell. And we call those specialized cells antigen-presenting cells. We call them APCs. And this cartoon is illustrating an example of the previous way of thinking about cancer vaccination, which is to get some of the tumor, a biopsy specimen from a patient, mix it with the right types of blood cells that can make these antigen-presenting cells, and then give them back to the patient as an injection as a way to vaccinate the patient. The idea being to get the immune system to recognize the cancer in the right way. And this process is very inefficient, getting the cancer to be found on the antigen-presenting cells that would stimulate the T cells. And we thought, well, one of the important points I mentioned about these macrophage cells is that they are these antigen-presenting cells. They can do the job of getting the T cells revved up. So now, what if we could take a patient's cancer, in this case, the leukemia, and directly convert it into those macrophage-like cells, those antigen-presenting cells, now this could be a very much more potent way of getting the T cells to be turned on in the right way to kill the cancer. And that was the idea that we came to after making the observation that we could convert leukemia cells into these antigen-presenting cells. And so we did a bunch of tricks to try to explore this by going into a system in mice that allows us to utilize an intact immune system. The previous experiments I showed you where the macrophage cells didn't cause cancer anymore were in mice where we put human cells into mice that had no immune system. But here we're looking at a situation where the mice have immune systems. And so this is, again, the experimental system. We could convert the cells into those macrophage-like cells. They have the features physically. This is a microscopic image. They look like macrophages. The things that are on macrophages go up over the time while we're getting the cells converted. The things that are on the B cell leukemia cells go down over time. And here is just another schematic showing all the different features. These are 5 different features that are critical for the antigen-presenting cells to do their job of revving up the T cells. And they're all shown here in the red boxes, is that using the methods that we developed, we could convert the cancer cells into these antigen-presenting cells. And then we wanted to do an experiment to see whether or not they would actually make the T cells respond. Like, do they do the job that we were hoping they would do? And the way we measure that is by mixing some T cells with those antigen-presenting cells. And we're looking at the portion of this of the cells in this plot that then fall in this region here. This is the positive control. If I tell my graduate students one thing that they got to learn while they're in grad school is how to design a positive control. It's back to the good old days of middle school science fairs. You have to know how to interpret your data. And the red here is really the experimental condition. What we found is the cells that we could convert into macrophages were able to stimulate the T cells in a way that the non-converted cancer couldn't. So this is suggesting we're on the right track, that our idea could work. And now this is really the key experiment. So here we're looking at mice that have no immune systems, and the purple is what you really want to look at. That's the condition that is the experimental condition. And in the absence of an immune system, some of the cells will convert into those macrophages, but not all of them. And so there's a little bit of a delay in the amount of time it takes for the leukemia to kill the mice. But if we do this in mice that have a fully intact immune system, they completely clear the leukemia. So the leukemia is converting into these macrophage cells that then tell the T cells, kill the leukemia. And they can do it. And we know that this develops the memory function of the immune system, right? When we get our vaccine or we get infected by something, the immune system has a memory component to it. And so now we take these mice that clear their leukemia and when we re-challenge them with leukemia, they don't develop leukemia. They have the function of immunological memory. And if we use a trick here in the green and the purple, and we get rid of the T cells that are in those mice, they no longer can cure themselves of that leukemia. So it's dependent on the T cells and it has immunological memory. This is another experiment to demonstrate the idea of immunological memory. What we do is we put on 2 flanks of the mice the cells that can convert and cells that can't convert. And so I'll just bring your attention to this side of the experiment. Every time the cells that can convert to those macrophages, when those tumors regress, the other side also regresses. So the immunological memory is systemic. It's circulating through the animal. It's not just present in one location. And then we wanted to ask ourselves, I'm going to skip this. It's a complicated experiment. And we're going to ask ourselves whether this could work in non-leukemia cancers. What about breast cancer or sarcoma cancer? In this case, this is a sarcoma. This is a breast cancer. This is another type of sarcoma. And a portion of those cells can also be converted into these macrophages. And when we do that experiment, we find that they all have survival benefits in mice. In fact, in some of the cases, we could even cure the sarcoma, which is a very uncurable type of aggressive cancer. So this lineage conversion, the ability to convert one cell type into another, one cancer type into these macrophage cells that can stimulate the immune system seems to extend beyond the blood system, although we have certainly studied it to less of a degree here. And then we wanted to go back to the patients that we had originally started studying, and we can collect from their bone marrow, their leukemia cells, and convert them to those macrophages, or we could, and at the same time, we can collect their blood and purify their T cells. So now we want to see if the T cells from the patient, which normally aren't doing anything against their leukemia, if we convert them to the macrophage cells, now will those T cells come back and hit that leukemia? And we haven't been able to get enough of these cells and enough of these patients to do the experiment in the mice I described, but this is just a measure of the T cells becoming activated, and they show increased levels of activation when we do this macrophage function. So suggesting that maybe this could actually work in patients, they might have T cells that could be active in clearing their tumors. So I'm happy to take some questions. I'll just stop there and leave you with these summaries. And I have a big group of scientists who work with me, many current, many post. This was a big party we had in the summer, but most of this work was done by a really talented Stanford PhD student, Miles Lindy, who did a lot of this work.
I come here, otherwise the people on Zoom cannot hear me.
Yeah.
At one point, you mentioned a cancer vaccine developed at Stanford,
Yeah.
20 years ago. So,
Yeah.
can you give us a historical perspective of cancer vaccines?
Cancer vaccination.
People like me didn't even know there was one.
Yes. Well, it turned out that it had only moderate efficacy. It's no longer on the market. I think that company ultimately abandoned that approach for more new and modern approaches. And with modern cancer immunology, there are many cancer vaccination approaches that are now ongoing and in clinical trials. Several have shown really exciting results just most recently at some of the national and international meetings. And again, it's this idea of getting the immune system revved up to fight off the cancer. As cancer develops, it doesn't develop in a vacuum. It co-evolves with the immune system, and it develops in a way that it can avoid immune recognition. That's natural selection right there at its finest inside one individual. So understanding how to get the immune system revved up to target the cancer is a long-standing goal of cancer immunotherapy.
So if you can manage that cell conversion, is that permanent? Or can it revert?
Yeah, that's a great question. The question was if you can, yeah, absolutely. The question was, is the cell conversion permanent or can it revert? With the patient samples and cells that we converted, we never found them to revert. But I wouldn't say that we have really extensively characterized that. And I think that's an area that we need to study more. And one of the ways you can deal with that is you can manipulate the cells outside the body in such a way that they can change to these converted macrophages, and you can treat them in such a way that they could never grow again, but they can still do their immune function. I know that sounds a little crazy, but that's actually how it works. So that's how the original vaccine worked as well.
Thank you.
Yeah. Yes.
How much of the personalization do you need for them to do that clinical thing?
Yeah, the question is how much personalization would you need to develop the macrophage? It's another great question. This entire approach would have to be one, if you're going to take the cells out of the body, manipulate them, and then give them back, that would have to be done on a patient by patient basis. But that's not bad in the sense that it could be done, but it's also not very scalable, right? To imagine having to do that for every single patient that you'd want to treat. So we are trying to figure out if there are any ways that we can cause the cellular conversion directly in the cancer itself, directly in the patient itself. But that's a little bit science fiction-y at this point. But okay, that's why they call it research. We'll work on that too. Yeah, please.
Yeah. So in this process, you're changing the patient's macrophages, but do you have to treat the patient with drugs to like interferon to eliminate their bone marrow before you
Yeah. The question is, in this approach, do we have to treat the patient with things like interferon to clear the bone marrow in order to be able to give back the macrophages? Did I capture the question correctly? Yeah. I think in that case, most immune responses take time to develop, right? That's why we got to get vaccinated, and you're not kind of in the clear for 6 weeks until after you've had your vaccine or your booster. So the idea of using a cancer vaccine definitely would be in a situation where the bulk of the tumor or the bulk of the leukemia has been cleared by something else, whether it be chemotherapy, surgery, radiation, depending on the particular cancer. And now you have a low level of disease that's left, and we know it's left because if we didn't treat the patients further, they would relapse, and then we'd have to deal with that from a clinical point of view. So the idea for cancer vaccination is to really use it at a time where the burden of cancer in the individual patient is lower.
Can I just ask, Ravi, can you explain the concept of immunological memory a little bit?
Yeah, the immune system. The question is, can I explain the concept of immunological memory? Yeah. When we encounter, our immune system encounters a pathogen of any type, we develop a memory for it over time. Like if we re-encounter it, then we have some immunity against it. And you can see that in individuals. I mean, we're also familiar with COVID in our lives. Just the last few years, people that didn't get vaccinated or got vaccinated, the more times you see it, the more your immune system is able to fight it off. And that is immunological memory. And there's a well understood set of mechanisms by which immunological memory works. So for example, if we, for the first time ever, encounter a pathogen, we make antibodies right away that are kind of low in their affinity and their potency. But over time, the immune system gets trained to make ones that are more potent, higher affinity. That type of thing is immunological memory.
Yeah, I had a question about machines. I'm always curious about the machines that are in the lab,
Oh, sure.
how influential they are. I mean, is there a new generation of machines that enable you to do this kind of research?
Yeah, that's a great question. The question is about the types of technology, the machines that we use in the lab. Are there technologies that enable us to do this type of research? The answer is yes. A lot of what I've shown were little plots that were made from flow cytometry. Flow cytometry is a technology that was invented at Stanford and commercialized now for the last 50 or 60 years, that allows you to interrogate individual cells for many of their different features. But of course, like all technology, it's advanced quite far, and we use that technology in many ways to allow us to be able to analyze the cells. As it relates to how we might think eventually of turning this into a therapeutic strategy, I think we might, we all got our COVID vaccines, hopefully, and the way we got them was RNA based, right? And the RNA is not naked, and meaning it is not just RNA in that shot. The RNA is encapsulated in something called lipid nanoparticles, which allow the RNA not to be destroyed by the environment, and that it can persist in your body and get to the right places. And lipid nanoparticle technology is advancing immensely, and ultimately we might want to try to cause the reprogramming in a patient by using RNA, that's the reprogramming RNA, in lipid nanoparticles. That's not machines, but it's definitely technology that we need.
Okay.
There were 3 questions on Q&A.
Oh, I can see them here. Let's see. First question is, I was wondering if any of this research and information on cells might have a connection to diseases affecting the spinal cord, such as multiple sclerosis and other diagnoses resulting in myelin degeneration. That's an interesting question. I think our work is really focused on cancer, but the idea of reprogramming cells into different fates is an important one for regenerative medicine, generally speaking. If you think about diseases in which organs or cells are gone, like multiple sclerosis, where the cells that are involved in making the myelin are gone, well, you got to figure out how to get those cells back. And you can get them from transplantation. Maybe you could get them by reprogramming other cells that would then take on these functions. That's an area of active research, but not one that is part of our work. The next question, what is it allowing these parts of cells to convert? Has there been a pattern of what parts of cells converting? Well, the question is really that we have started to understand that there are key factors that are lineage defining factors, that may be the best way to say it, and that by introducing them in an experimental way into other cells, you can force that cell to take on the other cell fate. The next question, have there been incidents of cancer adaptation and recurrence where the cancer becomes resistant to the created immune response? The answer is yes, yes, and yes. There are many different types of immune stimulating therapies that we use in cancer care today, everything from bone marrow transplant to different types of immune inhibitors. And most cancers ultimately do become resistant, and trying to understand the mechanisms of those resistance and how to avoid that is an area of great investigation across the broad biotech sector. Next question, would it be required to take the vaccine yearly until the cancer is completely destroyed? That's an interesting question. I think it would likely require multiple administrations of the vaccine, but ultimately to figure out how and when and the timing, that's going to require a lot of trial and error and eventually clinical studies. But cancer vaccines are coming, but the question is how effective they will be, and that remains to be seen.
Thank you.